to show what traits you'll get. So this one says, assume that tall is dominant over dwarf keys, and work out across with these different conjunctions, or, I don't know, different organisms. So there's one from a parent, there are two from parents, one's from mom, one's from dad, and heterozygous means that they're different, so it'll be like this. It'll be a big letter and then a little letter. And it doesn't matter what letter you use, but you want to make sure you use one that you can tell a difference. And then homozygous means that it'll be the same, so either big, big, or little, little. And these things, the letters, are called genotypes. And then your tall and dwarf are your phenotypes, which means that's their physical appearance. So for one, you'll have heterozygous tall, which means this, and you'll put it like this. And then dwarf, which means this, so you'll put that one on the side. And because these aren't sex-linked, it doesn't really matter which one's the mom or the dad. So I always put the big letter first, so D, D, and you just cross between these two. So then you do it with these two. D, D. And then again, the big letter first. D, D. And then D, D. And so this means you'll have a heterozygous tall, dwarf, dwarf. So it would be 50% hetero, tall, and then 50% dwarf. Number two. Because it's 2 dwarf, you'll have little d, little d, little d, little d, and you just go down again. And so you'll have 100% dwarf. For number 3, you have a homozygous tall, so it'll be big D, and then another dwarf, so little d, little d. And then you do big letters first. And you'll have 100% heterozygous tall. And that's it.
Okay, so our problem is guinea pigs may be yellow, white, or an intermediate cream color, so we're going to draw a cross diagram with yellow and white and then intermediate cream. So to begin, if we want yellow and white, we're going to have to make them different colors. So we're going to have yellow on top, and we're going to have white on the side. Okay, so we're just going to like match them up, so we'll go left and up, and then again left and up. And then we're going to go down and just keep going. So that these are our 4 outcomes. And then for this one, we're going to have 2 intermediate creams. So if they're intermediate, it means they're mixed between white and yellow. So we're going to do a white yellow on top and a white yellow on the bottom. That looks really bad. And then we're just going to cross it again. Y, W, Y, Y. So for this one, because they're W's and Y's, it's going to be 100% intermediate. And then this one has a WW, so it's going to be 25% white. And it has one YY, so it's also going to be 25% yellow. And then we have 2 that have Y and W in them, so this 50% will be cream, because these colors mix.
In guinea pigs, black and short-haired is dominant to brown and long-haired. Carry out a dihybrid cross to the F2 generation for a homozygous black short-haired and the other is brown long-haired. So dihybrid crosses are crosses between 2 different traits. Notice black, the color, black and brown, and then the hair type. So for these, you have to foil. So to foil, you go first, inside, outside, last. And because all these would be the same, you would get 4 big D, you just use the 1. And then the next one is brown, long-haired. So brown is always going to be B, B. And then long-haired is always going to be little d, little d. And so it happens the same way, and so you'll have the same thing 4 times if you do it all. So you just use the 1. And then you do a cross between these 2. And I always put the big letters first, so B, B, D, D. And then this is the F1 generation. After you get the F1 generation, you have to foil it again. So you'll get B, big D, and then you'll get big B, little d, and then B, big D, and then B, little d. And so then you do a cross between these.
And then you go through
to find out the phenotypic types. And so this one would be black and short hair, and this one would be the same thing, black and short hair. And then you just keep on doing that. Like this one would be black and long haired, this one would be brown and short haired, and then this one would be brown and long haired. So you just go through and find out all the different phenotypes, and that's it.
So this is a codominance problem, and it's in cattle there are red coats, which is RR, and white coats, which is R prime, and roan, which is R prime. And a roan means that it's red with white spots below. So if we start off with two red parents, you have RR and RR. So if you match them all up, you get RR for all of these, and it's 100% red. Grown parents, and you would have you have R, and then R prime matched with R and R prime. And when you do this, you get two big R's, and then R and R prime, then R prime and R, and then R prime and R prime. So because of this, you would have 25% red, 50% roan, and 25% white.
These are the sex-linked genotypes for males and females. Males are in green, and females are in black. So the normal genotype would be X big R for females, and for male it would be X big R and Y. For the carriers, it would be X big R and X little r for female, and males can't be carriers because they only have one X. And carriers have the dominant and recessive gene, but only express the dominant one. And then affected, they both have the recessive for the X's, so it would be X little r, and the male would be X little r and Y. A normal vision mother whose mother was colorblind has a son. Nothing is known about the color vision of the father. What is the probability the son will be colorblind? So because the mother's mother, the son's grandmother, was colorblind, the son's mother will be a carrier because a child will get an X, a female child will get an X from mom and an X from dad. So the mother's genotype will be X big R, X little r. So she's still normal vision, but she's a carrier. And since there's nothing known about the genotype of the father, we're just going to put him as X question mark Y. Sons only get a Y from the father, so it doesn't necessarily matter what the father is in this case. So you cross it down. See, these are the two sons. So the probability that the son will be colorblind is 50% because there's a chance for one son to be colorblind and one chance for a son to be normal.
So this is how regular DNA works. DNA has two strands, and they're joined together by base pairs. T goes with A goes with T, C goes with G, and G goes with C. T goes with A because they only have 2 hydrogen bonds, and C goes with G because they have 3 hydrogen bonds, so they can only join with those pairs. So T stands for thymine, A stands for adenine, C stands for cytosine, G stands for guanine, and U stands for uracil. So the top row is the original DNA that's written in black, and then the one written in green on the bottom is the mRNA. So T goes to A goes to U, C goes to G, and G goes to C. And then that pattern continues.
So this is the pedigree, and the shaded shapes are those that are affected, and the half shaded means they're carriers. So also, the squares mean it's a male, and the circles mean it's a female. So here we have an affected male married to a non-affected female, and they have 3 carrier daughters and 2 non-affected sons. Okay, so are these genotypes?
Hey, it's recording.
Cytosine is changed to guanine,